These are the astronauts, mice, and isolated test crews who gave scientists the first real evidence that deep space quietly rewires the human brain. This video walks through exactly what researchers have measured so far, using published studies from NASA, the European Space Agency, and independent university labs, so you can see the actual evidence instead of the usual space hype. If you're into this kind of deep, evidence-based look at what space travel really does to us, tap that like button, hit subscribe so you don't miss the next one, and drop a comment telling me where in the world you're watching from and what you're doing while you listen. I read every one of them, and I'm always curious who's out there at 2 in the morning learning about cosmic radiation with me. Alright, let's get into it. For most of human history, gravity was not something anyone had to think about. It simply held everything in place, the way it always had, and the brain built its entire understanding of up and down and where my body is in space, around that single, unchanging fact. Then, starting in the 1960s, a small number of people left the planet, and gravity stopped being a given. What nobody expected was just how much that single missing force, combined with a second invisible threat called cosmic radiation, would end up changing the physical shape and internal chemistry of the human brain itself. Not metaphorically, literally, measurably, on an MRI scan, in ways scientists are still working to fully understand. Here's the thing that makes this story different from most space content you've probably seen before. Earth doesn't just give us gravity, it also wraps us in something called the magnetosphere, an invisible magnetic shield generated by the planet's core that deflects the majority of dangerous cosmic radiation before it ever reaches us. Astronauts aboard the International Space Station, orbiting at roughly 400 kilometers up, are actually still tucked inside that protective bubble for most of their mission. The Apollo astronauts who went to the moon left that bubble entirely, venturing into fully unshielded space, but their round trips lasted only about 2 weeks, which kept their total radiation exposure relatively low. A mission to Mars is a completely different proposition. With current propulsion technology, just the one-way trip takes somewhere in the range of 6 to 9 months. And when you factor in time on the Martian surface and the return journey, the entire round trip is estimated at 2 to 3 years. For that entire stretch, the crew would be fully outside Earth's magnetic protection, exposed around the clock to a kind of radiation environment no human being has ever experienced for anywhere close to that long. NASA takes this seriously enough that the cosmic radiation threat to the brain has its own internal designation. They call it Risk 29, and it sits among the agency's most pressing concerns for any future crewed Mars mission, mainly because there is currently no shielding solution light enough to fly that can fully block galactic cosmic rays. And that's just one piece of the puzzle. On top of the radiation problem, there's microgravity itself, quietly reshaping the physical structure of the brain, chronic sleep loss chipping away at cognitive sharpness, and the psychological weight of total isolation, stacked together for years at a time with no way to simply step outside for fresh air. Scientists studying all of this are admittedly candid about where the field currently stands. Research into how spaceflight affects the brain specifically, as opposed to the rest of the body, is still, in the words of researchers publishing in the scientific literature, in its infancy. But infancy doesn't mean nothing. It means the first real data points have started coming in, and they are genuinely fascinating, occasionally unsettling, and worth walking through carefully, one finding at a time. To really appreciate why the Mars timeline specifically matters so much to brain researchers, it helps to line it up against every other kind of human spaceflight that's come before it. The original Mercury and Gemini missions in the 1960s lasted hours, or, at most, a couple of weeks. Apollo lunar missions, the ones that actually left the magnetosphere entirely, were even shorter round trips, typically a matter of days from launch to splashdown. Even the ISS, humanity's primary platform for long-duration spaceflight research today, keeps its crews largely within the protective reach of Earth's magnetic field, despite missions now regularly stretching to 6 months and occasionally close to a full year. A Mars mission breaks that pattern completely, on every axis at once. It's longer than any previous human spaceflight by a wide margin, and for the entire outbound transit and the entire return transit, the crew would be fully exposed, outside the magnetosphere, in a way no ISS crew or Apollo crew has ever had to sustain. That combination, unprecedented duration and unprecedented radiation exposure happening simultaneously, is exactly why researchers keep describing Mars specifically, rather than the Moon or the ISS, as the scenario that turns every one of the risks in this video from a manageable inconvenience into a genuinely open scientific question. It also helps to understand just how new the underlying dataset really is, because the honest answer is that scientists simply haven't had that many long-duration human missions to study. Meaningful long-duration spaceflight, missions stretching months rather than days, has really only existed for a relatively short window of human history. A comprehensive 2024 review in The Lancet Neurology notes that the number of long-duration human spaceflights has increased substantially over roughly the past 15 years, which is precisely the window that has allowed brain-specific research to grow from a handful of speculative case reports into something closer to a genuine, structured field with repeated findings across multiple independent research teams. Before that window opened, researchers were mostly limited to short shuttle missions lasting a couple of weeks, nowhere near long enough to reveal the kind of gradual structural brain changes this video is about to walk through. It's only really since the International Space Station matured into a platform capable of supporting crews for 6 months, sometimes closer to a full year, that scientists have had enough astronauts, enough scan data, and enough accumulated mission time to start noticing real, statistically meaningful patterns rather than isolated anecdotes. It's worth being precise, too, about exactly which parts of the human body researchers are talking about when they say space rewires the brain, because that word can mean several genuinely different things, and this video is going to cover essentially all of them, one category at a time. Sometimes it means something as literal as physical shape, the brain's actual position and volume shifting inside the skull. Sometimes it means microscopic structural damage, individual neurons losing branching connections or having their protective insulation degraded. Sometimes it means functional performance, how quickly and accurately a person can think, remember, and react under real operational pressure. And sometimes it means something considerably harder to pin down on a scan at all, a genuine, lasting shift in outlook, values, and self-concept, the kind of change researchers can really only detect by asking people directly what changed inside them. All 4 of these categories show up in the published research, and treating them as separate, distinct threads, rather than blending them into one vague claim that space changes your brain, is really the only honest way to tell this story. And it's the approach this video is going to follow all the way through, one category, one study, and one researcher at a time. Let's start with the closest thing space science has to a landmark human experiment on this exact question, because it involves 2 brothers, one genome, and one very unusual scientific opportunity that NASA almost couldn't have designed better if they'd tried. In March of 2015, astronaut Scott Kelly launched to the International Space Station to begin what would become a 340-day mission, just shy of a full year in orbit. What made this particular mission scientifically priceless wasn't just its length, it was the fact that Scott Kelly has an identical twin brother, Mark Kelly, also a retired astronaut, who agreed to stay behind on Earth in Tucson, Arizona, and serve as a living control subject. Because identical twins share essentially the same genetic starting point, comparing Scott's body and brain to Mark's over that same stretch of time gave researchers something they almost never get in human biology, a way to isolate the effects of the space environment itself, largely stripped of the usual genetic noise that makes comparing 2 different people so messy. There's a reason twin studies carry so much scientific weight in the first place, and it's worth pausing on before getting into what researchers actually found. Comparing 2 unrelated people is inherently messy, because any difference between them could be explained by countless factors that have nothing to do with the thing you're actually trying to measure. Different genetics, different upbringing, different baseline health, different everything. Identical twins largely eliminate that noise, since they start from essentially the same genetic blueprint. When one twin spends nearly a year in space while the other stays on the ground living an otherwise ordinary retired life in Arizona, any meaningful biological divergence that shows up between the 2 becomes far easier to attribute specifically to the space environment itself, rather than to some unrelated difference the 2 men happen to be born with. NASA has described this setup as an exceptional, close to unrepeatable scientific opportunity, given how rare it is to have two identical twins who are both trained, flight-qualified astronauts in the first place. It's also worth noting this wasn't purely a simple nature versus nurture comparison. Epigenetics, the study of how environmental conditions can switch genes on or off without altering a person's underlying DNA sequence, played a central role in how researchers interpreted their findings, since so much of what changed in Scott Kelly's body involved shifts in which genes were actively being expressed, not changes to his fundamental genetic code itself. NASA didn't run this as one tidy little experiment either. 10 separate science teams from around the country signed on, coordinating together as one integrated research group, in what became known simply as the Twins Study. Over the course of 27 months, spanning the period before, during, and after Scott's mission, researchers collected more than 300 biological samples from the two brothers. Blood, saliva, urine, stool, you name it, all shipped back and forth and analyzed by labs using some of the most advanced molecular tools available at the time. The full results were finally published in April of 2019 in the journal Science, and reading through them, you start to understand why NASA calls this a landmark study. Some of what they found was almost eerie in its precision. Christopher Mason, a functional genomicist at Cornell University who led part of the genetic analysis, described watching Scott's immune-related genes shift into overdrive during spaceflight, almost as though his body sensed it had entered a completely foreign environment and needed to prepare for a fight it couldn't quite name. Mason put it simply, the immune system in space goes on what he called almost a high alert, trying to make sense of the new environment around it. Scott's chromosomes told an even stranger story. Researchers documented actual structural changes in his chromosomes during the mission, with segments getting swapped, inverted, or even merged together, the kind of chromosomal rearrangement that in other contexts has been linked to infertility or certain forms of cancer. Most of these shifts weren't permanent. Within about 6 months of Scott's return to Earth, the vast majority of his altered gene expression had reverted back to pre-flight levels. But not all of it. Roughly 7% of his genes stayed expressed at different levels even after that 6-month recovery window closed, and a good number of those persisting changes were tied specifically to DNA repair and immune function, meaning some part of what space did to Scott Kelly's biology simply didn't bounce back. It's worth explaining a little more clearly what a chromosomal rearrangement actually is, because the phrase can sound abstract without context. Every one of your cells carries chromosomes, tightly packaged bundles of DNA, and under normal circumstances, those chromosomes stay structurally intact and consistent throughout your life. A rearrangement means a piece of one chromosome has physically broken off and been reattached somewhere else, sometimes in reverse order, sometimes fused onto an entirely different chromosome altogether. These events aren't unique to spaceflight. They happen at some low background rate in ordinary human cells throughout a person's life, often triggered by exposure to things like radiation or certain chemicals, and the body typically has repair mechanisms designed to catch and fix this kind of damage before it causes problems. What researchers found notable in Scott Kelly's case wasn't that this kind of rearrangement happened at all, but that it happened at a rate researchers considered significant enough to document and publish as a distinct finding of the mission. The two categories of downstream risk associated with this kind of chromosomal disruption, infertility and certain cancers, are exactly why researchers treat this finding as one of the more genuinely consequential results to come out of the entire Twins study, even though, as the reassuring framing from NASA and the National Institute on Aging emphasized, Scott Kelly's overall health was described as mostly sustained by the time all the data had been analyzed. Now here's where the story gets genuinely interesting for anyone curious about the brain specifically, and also where it gets a little murky, in a way that's worth being honest about, rather than smoothing over for the sake of a cleaner narrative. As part of the Twins study, researchers ran Scott through a battery of cognitive tests measuring things like short-term memory, speed, accuracy, and logical reasoning, comparing his results to his own pre-flight baseline and to Mark's results on the ground. One widely reported finding from that testing showed Scott's speed and accuracy on memory and logic tasks had declined relative to where he started. NASA itself was careful to note that whether this decline can be definitively pinned on spaceflight is unclear, partly because of limitations built into how the observations were structured. That's an important qualifier, and it's one NASA made themselves, not something critics tacked on afterward. At the same time, a separate NASA-affiliated summary of the very same study described something almost contradictory on the surface. According to that account, with a few exceptions, Scott's broader cognitive performance, things like mental alertness, spatial orientation, and his ability to recognize emotional expressions, remained largely unchanged throughout his time in space compared to his brother back on Earth. So which is it? Did his cognition decline, or did it hold steady? The honest answer is both accounts are legitimate, and the apparent contradiction says something important about how complicated brain research in space actually is. The full cognitive battery covered multiple distinct mental domains, and it's entirely possible for memory and logic processing speed to dip slightly, while spatial orientation and emotional recognition stay rock solid. Real science, especially when you're working with a single human subject, rarely hands you one clean, simple verdict. It hands you a mixed bag of results across different measures, and the responsible thing to do is present that mixed bag honestly, instead of flattening it into a punchier headline. There's a broader lesson buried in that apparent contradiction worth carrying forward into the rest of this video too. The twins study, for all its genuine scientific value, was still fundamentally a study of one person compared to one other person. That's an extraordinary setup for isolating variables, given how rare identical twin astronauts are. But it also means the cognitive findings from this particular study carry an inherent limitation that a larger study with dozens or hundreds of subjects wouldn't have. A single individual's test scores can shift for all sorts of ordinary reasons having nothing to do with spaceflight at all. Mood on a given testing day, fatigue, unfamiliarity with a specific test format, simple measurement noise. NASA's own hedging language around the memory and logic findings, explicitly noting it's unclear whether the decline can be definitively attributed to spaceflight, reflects exactly this kind of appropriate scientific caution. It's a useful reminder to keep in mind as we move into research involving larger groups of astronauts and larger groups of test animals, where researchers can rely on statistical patterns across many subjects rather than a single, if scientifically extraordinary, pair of twins. Beyond the cognitive testing, the twins study reinforced something researchers had suspected but hadn't confirmed with this level of molecular detail before: that a human body can adapt to, and largely recover from, nearly a year in space. The overall framing from NASA and the National Institute on Aging, which helped fund parts of the analysis, was that Scott's health was, in their words, mostly sustained throughout the mission. That's genuinely reassuring context to hold on to as we move into some of the more unsettling findings ahead, because it's a reminder that the human body, brain included, is far more resilient and adaptable than you might assume after hearing about chromosome rearrangements and cognitive decline. One more detail from this mission is worth sitting with before we move on, mostly because of what it says about the psychological weight of the environment Scott was living in for nearly a year. Across those 340 days, according to information NASA itself released, Scott Kelly came into physical contact with only about 12 other human beings, total. Not 12 people a week. 12 people across the entire 11-month mission. Think about what that actually means for a moment. Nearly a year of your life, spent almost entirely surrounded by the same small handful of crewmates, floating 400 kilometers above everyone else you've ever known, with Earth itself reduced to a bright blue curve outside a small window. We'll come back to what that kind of isolation does to the mind later in this video, because it turns out to be its own distinct area of research, separate from anything happening at the molecular or structural level in the brain. But keep that number in the back of your mind, 12 people in 340 days, because it sets the stage for everything that follows about a Mars mission that would need to stretch that kind of isolation out 3 or 4 times longer, with no possibility of an early return if things go wrong. The twins study gave us our first serious human data point, but to really understand what deep space might do to the brain specifically, over a mission length even longer than Scott Kelly's, scientists have had to turn to a different kind of subject entirely, one that lets them study effects an ethics board would never approve testing directly on a person. That subject is the laboratory mouse, and the radiation research built around it is where this story takes a considerably darker turn. The scientist most associated with this line of research is Charles Limoli, a professor of radiation oncology at the University of California, Irvine School of Medicine. Limoli's team has spent years using a facility called the NASA Space Radiation Laboratory, located at Brookhaven National Laboratory in Upton, New York, which is capable of bombarding living tissue with the same kinds of high-energy charged particles that make up galactic cosmic radiation in deep space. The idea is straightforward in concept, even if the science behind it is anything but simple. If you can't ethically expose a human being to years of unshielded cosmic radiation just to see what happens to their brain, you expose lab animals to a carefully calculated equivalent dose instead, and you study the tissue afterward. In October of 2016, Limoli's team published results that made headlines well beyond the usual scientific press, and the timing was almost cinematic. That same month, President Obama had publicly announced ambitions to send American astronauts to Mars by the 2030s, and here was a study suggesting the human brain itself might not hold up well for the trip. Limoli's mice had been exposed to a radiation dose designed to simulate what a crew would encounter on a 6-month, one-way journey to Mars. What his team found was significant: long-term brain damage, including cognitive impairments and dementia-like effects, driven by inflammation in brain tissue and direct structural damage to individual neurons. This wasn't a subtle effect either. The new findings revealed a far greater extent of brain damage than an earlier, shorter 6-week pilot study the same team had run the previous year, meaning the damage appeared to compound the longer the radiation exposure continued, rather than staying flat. When researchers looked closely at the animal's brain tissue under imaging, they found a sharp reduction in dendritic complexity, the intricate, branching, tree-like structures that neurons use to physically connect with one another and pass signals back and forth. Fewer, simpler branches generally means a less capable network, less able to form and hold on to the kinds of connections that underlie memory and learning. Limoli didn't mince words when reporters asked him what this meant for a future Mars crew. His quote has been repeated in coverage of this research ever since. "This is not positive news for astronauts deployed on a 2 to 3 year round trip to Mars." Researchers went further, noting the damage they observed appeared likely to affect memory specifically, and could plausibly heighten anxiety and fear responses as well, based on how the animals behaved after exposure. And crucially, this was the second study out of Limoli's lab to reach a similar, sobering conclusion. 2 separate rounds of testing, using 2 different exposure protocols, both pointed toward cosmic radiation causing what looked like permanent, and likely untreatable, damage in the mouse brain, with the specific structures affected bearing meaningful similarity to corresponding structures in the human brain. It's worth flagging one adjacent detail here for context, even though it's a separate line of research rather than a direct extension of Limoli's work. An earlier 2013 study in mice had already suggested that radiation exposure might accelerate the development of Alzheimer's-like changes in the brain, hinting that whatever damage cosmic rays do isn't necessarily a totally novel category of harm, but might overlap mechanistically with processes we already associate with age-related cognitive decline here on Earth. That overlap turns out to matter a great deal, and we'll circle back to why in a moment. Limoli's team didn't stop in 2016. Real deep space radiation doesn't arrive in one dramatic burst the way early lab experiments simulated it. It's chronic, low-level, and constant, day after day, month after month, for the entire length of a mission. So in a follow-up study published in the journal eNeuro in 2019, and covered extensively by outlets including space.com, Limoli's team redesigned their approach specifically to mimic that slower, chronic exposure pattern rather than a single acute dose. The results held up. Once again, the animals showed serious memory and learning impairments. And once again, researchers observed increased anxiety and fear-like behavior in the exposed group compared to controls. Limoli's assessment of what this means for real mission planning was blunt. These chronic low-dose rate, low-dose exposure scenarios are going to increase the risk of developing, perhaps, mission-critical performance deficits. What exactly those are, we'll never know until we get out there. That last line deserves a moment of its own, because it's an unusually honest thing for a scientist to say on the record. We'll never know until we get out there. Despite years of careful, methodical rodent research, despite imaging studies and behavioral testing and dose calculations, there is still a hard limit to how much this line of research can tell us, simply because no human being has ever actually made that trip. Everything Limoli's lab has found is an extrapolation, an educated, evidence-based, carefully modeled extrapolation, but an extrapolation nonetheless. Limoli's group isn't the only one working this problem. A separate team of radiation biologists at Eastern Virginia Medical School approached the same basic question from a slightly different angle. Writing about their own findings in The Conversation, in a piece published in 2019, these researchers described exposing mice to 6 months of low-level neutron radiation, specifically chosen because neutrons are one of the fragmentation byproducts created when high-speed cosmic radiation particles collide with a spacecraft's hull, or with the human body itself. After that 6-month exposure period, the team examined the animal's neurons directly and found damage to some of the brain's most delicate microscopic structures: dendrites, the branching connectors mentioned earlier; myelin, the fatty insulation that wraps around nerve fibers and allows signals to travel quickly and efficiently; and synapses, the actual junction points where one neuron passes a signal to the next. Damage to any one of these 3 structures on its own would be a problem. Damage across all 3, simultaneously, in the same brain tissue, points toward a fairly broad disruption of the brain's basic wiring and communication infrastructure. This same Eastern Virginia team went a step further than most published radiation research, and did something genuinely striking. They took their rodent behavioral data and used it to calculate an estimated human risk figure for a real Mars crew. Their projection, in a crew of 5 astronauts making the full round trip to Mars and back, they would expect at least one crew member to display severe deficits in cognitive function by the time they return to Earth. It's worth being very precise about what that number actually is and isn't. It is not a measured outcome from any human mission, because no such mission has happened yet. It is a mathematical extrapolation, built from rodent brain and behavior data, translated into projected human risk using the researchers' own modeling assumptions. That doesn't make it meaningless. If anything, it makes it more useful as a planning tool, a rough early warning estimate mission designers can use to justify investing in protective countermeasures now, well before any crew actually launches. But it's a projection, not a confirmed medical finding about an actual human being, and treating that distinction carefully matters if we want to represent this research honestly. Underneath all of these rodent findings sits a fairly clear physical mechanism, and understanding it helps explain why cosmic radiation is such a uniquely difficult problem to engineer around. Cosmic radiation particles travel at speeds approaching the speed of light. When they collide with the metal hull of a spacecraft, or with living tissue inside a human body, that collision generates a spray of secondary particles, including neutrons, that carry enough leftover energy to keep tearing through the body. As these secondary particles interact with atomic nuclei inside cells, they generate free radicals, unstable, reactive molecules that go on to damage cell membranes, DNA strands, and other fine cellular architecture, brain cells very much included. This is part of why simple, thick-walled shielding doesn't fully solve the problem the way you might assume. Cosmic radiation is so penetrating that shielding thick enough to reliably block it would add an enormous amount of mass to a spacecraft, and every extra kilogram of mass is enormously expensive to launch and maneuver through deep space. NASA's own risk assessment work refers to this specific threat by that internal shorthand mentioned earlier, Risk 29. And some of the more dramatic press coverage around this research has gone so far as to raise the possibility, in the most extreme unmitigated scenario, of astronauts ending up with catastrophic neurological impairment. That framing leans more toward journalistic dramatization than a precise, peer-reviewed medical prediction, and it deserves to be flagged as such, rather than repeated at face value. But it does capture just how seriously the underlying research community takes this particular risk. It also helps to understand why the specific type of radiation found in deep space is so much harder to defend against than the radiation we're used to dealing with on Earth, or even in low Earth orbit. Medical X-rays, airport scanners, even the background radiation you absorb during a transatlantic flight are what physicists classify as relatively low-energy forms of radiation. the kind that ordinary shielding, a lead apron, an aircraft hull, handles reasonably well. Galactic cosmic rays are a different category of problem entirely. They're made up of high-energy, heavy-charged particles, flung across the galaxy by exploding stars and other violent cosmic events, and they arrive moving at a meaningful fraction of the speed of light. When something moving that fast slams into ordinary shielding material, it doesn't just get absorbed the way a slower particle would. It shatters the atoms it hits, creating that spray of secondary particles mentioned earlier, meaning in some cases, adding shielding can actually make the local radiation environment worse rather than better, by generating more of these secondary fragments right where the crew is sitting. That's part of why the solution to this problem isn't as simple as bolting on thicker walls, and why NASA continues treating it as one of the hardest unsolved engineering and biological challenges standing between where we are now and a real crewed Mars mission. There's also a broader body count worth mentioning, because the brain isn't the only organ researchers have flagged as vulnerable to this same category of deep space radiation. A separate study, unrelated to Limoli's brain work but conducted around the same period, found evidence that cosmic radiation exposure during the Apollo missions may have contributed to cardiovascular damage in some of the astronauts who flew beyond the magnetosphere on their way to the moon. That finding isn't a brain-specific result, but it's worth including here as context, because it reinforces the same underlying point from an entirely different angle. Once a human body leaves the protection of Earth's magnetic field, cosmic radiation doesn't selectively target one organ. It moves through tissue indiscriminately, and the brain, the heart, and likely other organs besides, all sit somewhere on the list of things researchers now believe deserve serious, dedicated protective attention before anyone attempts a multi-year deep space mission. It's worth explaining a little more about how researchers actually calculate the radiation dose they give these test animals, because the whole point of this research only works if the exposure genuinely mirrors what a real Mars crew would experience. Limoli's team specifically designed their dosing to simulate the radiation environment of a 6-month, one-way transit to Mars, using charged particles matched as closely as possible to the composition of real galactic cosmic radiation, not a generic stand-in radiation source. That distinction matters because different types of radiation damage biological tissue in meaningfully different ways, and a dose calibrated to mimic a routine medical X-ray would tell researchers essentially nothing useful about what astronauts will actually encounter once they're fully outside the magnetosphere. The behavioral changes researchers documented in these irradiated animals went beyond simple memory and learning tests too. Exposed animals showed patterns consistent with heightened anxiety and increased fear responses, changes researchers were able to measure using standardized rodent behavioral assays designed specifically to detect exactly this kind of shift in emotional processing. Put together, the picture that emerges isn't just radiation makes remembering things harder. It's a more complete portrait of an animal whose entire emotional and cognitive baseline had shifted, becoming both less capable of learning new information and more prone to fear and anxious behavior at the same time. A combination that, if it translates to humans in any meaningful way, carries obvious implications for how well a crew could function together and make sound decisions during a multi-year mission with no possibility of turning back. There's also a meaningful difference between how Limoli's earlier 2016 work and his later 2019 follow-up study delivered radiation to their test subjects, and that difference is part of why the 2019 results carry particular weight for real mission planning. The original studies used what researchers call acute dosing, delivering the full simulated radiation exposure over a relatively short, concentrated period, which is easier to control in a lab setting but doesn't perfectly match how radiation actually accumulates during a real mission. Real deep space radiation arrives gradually, in a slow, continuous trickle over months, not in one dramatic burst. So Limoli's team deliberately redesigned their 2019 study around chronic, low-dose rate exposure instead, spreading the same overall radiation dose out gradually to better match real mission conditions. When the results held up under this more realistic exposure pattern, that meaningfully strengthened the case that this isn't some artifact of an unrealistic lab shortcut. It's a finding that survives a serious attempt to make the experimental conditions as true to life as a ground-based lab possibly can. Not every scientist working this problem believes the story ends with simple, straightforward neuron death either, and that distinction actually opens up some genuine reason for optimism. Richard Britton, another researcher at Eastern Virginia Medical School, received roughly $1.2 million in dedicated research funding, part of a much larger multi-institution effort, specifically to study the biochemical mechanisms behind radiation-induced cognitive impairment in irradiated rats, including detailed proteomic analysis, meaning a close look at exactly which proteins inside brain cells are being disrupted by radiation exposure. Britton's working hypothesis is that a lot of the cognitive impairment researchers are seeing doesn't come purely from neurons simply dying off, but from more complex and potentially reversible biochemical processes going wrong inside cells that are still very much alive. His own words capture the hopeful angle buried inside all this otherwise sobering research. Once we understand what's not working, then maybe we can fix it. And there's a genuinely useful piece of overlap worth mentioning here too. Because radiation-induced brain damage appears mechanistically similar to the kind of free radical injury associated with natural aging and neurodegenerative diseases like Alzheimer's here on Earth, this entire line of space radiation research carries a plausible secondary benefit. Whatever protective treatments scientists eventually develop to shield an astronaut's brain on the way to Mars could end up informing how we treat age-related cognitive decline in people who never leave the planet at all. Britton's proteomic work is only one piece of a much larger, coordinated effort tackling this exact problem, and it's worth understanding the scale of that effort to appreciate just how seriously the research community takes it. Reporting on this area has described a total of 12 separate research teams working in parallel, each investigating a distinct angle of the same underlying cosmic radiation risk, all contributing pieces toward NASA's larger goal of eventually determining a maximum safe cosmic radiation dose for the human brain, and mapping out exactly how radiation produces cognitive impairment at a mechanistic level, precisely so that targeted biological countermeasures can eventually be developed and deployed before any real crew commits to a multi-year deep space mission. That kind of parallel, distributed research structure, a dozen teams chasing different angles of the same problem simultaneously, rather than one lab working in isolation, is fairly typical of how a genuinely difficult, high-stakes scientific problem tends to get approached, and it's a useful reminder that everything covered so far about cosmic radiation and the brain represents an active, ongoing, heavily resourced research push, not a single isolated study sitting on a shelf somewhere, waiting to be noticed. Right now, engineers and biologists are working on this problem from multiple angles simultaneously. And it's worth understanding the actual menu of options on the table, because none of them are simple. The first approach is passive shielding, essentially building thicker protective layers, sometimes incorporating water as part of the shielding material, either around the entire spacecraft, or, more realistically given weight constraints, around a smaller dedicated sleeping or shelter compartment, where the crew could retreat during periods of heightened radiation. The logic behind that smaller shelter compartment version of the idea is fairly clever once you think it through. Rather than trying to armor an entire spacecraft, which would be prohibitively heavy, mission designers could instead build one smaller, heavily shielded refuge inside the ship, sized just large enough for the crew to retreat into during periods of unusually intense radiation, such as a solar storm, while accepting somewhat lighter protection for the rest of the vehicle during normal, lower-intensity periods. Water is a particularly attractive shielding material for this kind of compartment, specifically because a spacecraft already needs to carry substantial water reserves for drinking and life support regardless, meaning that water could serve double duty as both a life support resource and a radiation shield, without adding as much additional deadweight mass as a purpose-built shielding material would. The problem, as already mentioned, is weight. Cosmic radiation is penetrating enough that meaningful protection requires a lot of mass, and mass is the single most expensive constraint in spaceflight. The second approach is active shielding, essentially generating an artificial electromagnetic field around the spacecraft designed to deflect incoming charged particles the same way Earth's own magnetosphere does naturally. It's an elegant idea on paper, and it sidesteps the weight problem of passive shielding. But the engineering required to generate and sustain a field like that around an entire crewed vehicle remains, as of now, unresolved. Part of what makes active shielding so much harder in practice than it sounds on paper is scale. Earth's own magnetosphere is generated by an enormous, planet-sized molten core that has been running continuously for billions of years, and engineers trying to replicate even a small fraction of that same protective effect around a single spacecraft are essentially trying to compress a planetary-scale phenomenon down into something that can be built, launched, and powered using current propulsion and energy technology, a genuinely difficult miniaturization challenge layered directly on top of the already difficult underlying physics involved. The third and newest approach is essentially pharmaceutical, developing medications capable of repairing radiation damage after it happens, protecting neurons proactively before exposure even occurs, or in the most ambitious version, actually restoring damaged neural structures after the fact. None of these three approaches has been finalized as the adopted solution for a real Mars mission. All three remain active areas of ongoing research, which is a fairly accurate summary of where the entire radiation and the brain question currently stands. Real, serious, well-funded science, chasing a problem that hasn't been solved yet. Radiation is only half the story though, and in some ways, it might not even be the half that affects the most astronauts on the most missions. Because long before anyone gets far enough from Earth to worry seriously about deep space cosmic rays, something else is already quietly reshaping their brain, and it's been happening to essentially every long-duration astronaut who has ever flown, right there in low Earth orbit, well within the protective magnetosphere. It's called the fluid shift, and it turns out gravity was doing far more structural work inside your skull than most people ever stop to consider. Here on Earth, gravity is constantly pulling fluid in your body downward, toward your legs and feet. Your circulatory and lymphatic systems evolved specifically around managing that constant downward pull. The moment a person enters microgravity, that pull essentially disappears, and without it, fluid that would normally sit lower in the body redistributes upward, toward the head and chest. Researchers call this a cephalad fluid shift, and it's become one of the most extensively documented physical changes astronauts undergo. You've probably actually seen a visible version of this effect, without realizing what caused it, in footage of astronauts whose faces look noticeably puffier once they've been in orbit for a few days. That same fluid shift happening beneath the skin on an astronaut's face is also happening, less visibly, but far more consequentially, inside their skull. A series of peer-reviewed neuroimaging studies, led by researchers including Donna Roberts and colleagues, and expanded on by numerous follow-up teams over the past decade and a half, have used pre-flight and post-flight MRI scans to document exactly what this fluid shift does to brain structure. The findings are remarkably consistent across multiple independent studies. The brain itself physically shifts upward within the skull. The narrow subarachnoid space at the very top of the brain, near the vertex, gets compressed and narrowed, as though the brain is being gently pressed upward against the inside of the skull. The brain's fluid-filled ventricles, the internal cavities that hold cerebrospinal fluid, expand in volume, and the cerebrospinal fluid itself gets redistributed throughout the skull in a measurably different pattern than it follows on Earth, including increased pulsation, sometimes called stroke volume, in a narrow channel called the cerebral aqueduct, along with reduced overall cerebral compliance, meaning the brain's fluid systems become less flexible in how they absorb normal pressure changes. There's an entire secondary system tied up in all of this too, called the glymphatic system, essentially the brain's built-in waste clearance and fluid drainage network, running through structures called perivascular spaces that wind alongside the brain's blood vessels. Multiple MRI studies have found these perivascular spaces measurably enlarge after long-duration spaceflight, particularly in the basal ganglia and in white matter regions. Interestingly, one detailed comparison study found this particular effect showed up more prominently in NASA astronauts than in Roscosmos cosmonauts serving aboard the very same space station, under broadly similar mission conditions. Researchers haven't fully pinned down why that difference exists, whether it traces back to genetics, training protocols, exercise regimens, or something else entirely, but it's a reminder that even within a shared spacecraft and a shared mission timeline, individual variation in how brains respond to microgravity is real and not yet fully explained. That same comparison study did find one point of clear agreement between the two space agencies' crews, though. Both the NASA and Roscosmos astronauts studied showed a similar degree of lateral ventricle enlargement and a similar decrease in the subarachnoid space at the very top of the brain, and that shared ventricle expansion was found to correlate directly with the degree of white matter perivascular space enlargement each astronaut showed. In other words, whatever nationality-linked or training-linked factor is driving the difference in basal ganglia and white matter perivascular changes, the more fundamental fluid shift mechanism connecting ventricle size to perivascular space size appears to hold true across both space programs, suggesting it reflects something more universal about how any human brain, regardless of training background, responds to an extended stretch of microgravity. Studies like this one, deliberately comparing crews from different space agencies flying the same missions on the same station, are quietly valuable in a way that's easy to overlook. Spaceflight research has historically been conducted somewhat separately by different national space programs, each running its own studies on its own astronauts, which makes genuine cross-agency comparisons like this one relatively rare and correspondingly useful whenever they do happen. Finding both a meaningful difference, the perivascular space discrepancy, and a meaningful point of agreement, the shared ventricle and subarachnoid space pattern, within the same single study, gives researchers a rare, direct way to start separating which effects of spaceflight on the brain are likely universal to human physiology in general, and which might instead trace back to something more specific about training, genetics, or national space program protocols, a distinction that matters quite a bit for planning any future mission that, like a real Mars expedition almost certainly would, brings together astronauts trained by different space agencies into one single, shared crew. The changes don't stop at fluid distribution either. Gray matter, the brain tissue responsible for most of its actual processing power, shows measurable volume decreases in specific regions after spaceflight, particularly around the frontal and temporal poles and near the eye sockets. At the same time, researchers have documented small, localized gray matter increases in the primary somatosensory and motor cortex, the regions responsible for processing bodily sensation and controlling movement. That combination, decreases in some areas paired with increases in others, is actually a meaningful clue about what's happening. Rather than simple, uniform damage, researchers increasingly interpret this pattern as evidence of neuroplasticity, the brain's underlying capacity to physically reorganize and strengthen certain connections in response to a genuinely new sensory and motor environment. In other words, at least some of what's happening isn't the brain breaking down. It's the brain adapting, quite literally rewiring parts of itself to handle moving, reaching, and orienting the body in a world with no reliable down. White matter, the brain's internal wiring made up of long nerve fiber tracts that connect distant brain regions to one another, shows its own distinct pattern of change. Researchers have documented altered volume and microstructural integrity across specific major fiber tracts, including the corpus callosum, which connects the brain's two hemispheres, the arcuate fasciculus, the corticospinal tract, several corticostriatal tracts, and multiple cerebellar tracts, along with the superior and inferior longitudinal fasciculi. One detailed 2019 study quantified the scale of these white matter changes fairly precisely, finding shifts ranging from roughly 0.75% to 1.25% across the affected tracts, alongside broader extracellular free water changes ranging from about 2.5% to 4% across different brain regions. Those percentages might sound small in isolation, but keep in mind these are measurable, statistically significant structural shifts happening inside a living human brain, detected using standard clinical MRI technology after a period of months in orbit. It's worth briefly explaining what researchers mean by extracellular free water, since that term shows up repeatedly across this body of research, and it's a slightly different concept from the ventricle and CSF changes already covered. Extracellular free water refers specifically to water sitting in the spaces between brain cells, outside the cells themselves, as opposed to water held inside cell membranes or flowing through the larger, more obvious channels like the ventricles. Tracking this specific kind of fluid gives researchers a more fine-grained picture of exactly where fluid is accumulating or draining away at a near microscopic level throughout the brain, beyond what's visible just by measuring the size of the large, easily seen ventricular cavities. One study using this specific measure found a significant, widespread increase in free water volume in the frontal, temporal, and occipital lobes after spaceflight, alongside a significant decrease in free water specifically in the posterior area near the top of the brain, the same region already noted as getting compressed by the upward shifting brain. That combination, increases in some lobes and decreases right at the vertex, paints a more detailed picture of fluid actively being pushed and redistributed around the astronaut's brain, rather than simply accumulating everywhere in a uniform way, which lines up with, and adds further texture to, the broader fluid shift story already covered. One of the most detailed studies in this entire area came out in June of 2023, published in Scientific Reports, and it's worth walking through in some detail because it directly addresses a question every future Mars mission planner desperately needs answered. Does this brain change keep getting worse the longer someone stays in space, or does it eventually level off? Researchers tracked 30 astronauts, divided into 3 groups based on mission length, roughly 2 weeks, roughly 6 months, and roughly 1 full year, comparing detailed MRI scans taken before launch against multiple follow-up scans taken after return to Earth. The astronauts on 6-month missions showed the greatest ventricular expansion of the 3 groups, with volume changes exceeding 0.75 milliliters. Here's the genuinely reassuring part of the finding though. Among astronauts who had spent a full year in space, that ventricular enlargement appeared to level off rather than continuing to climb, suggesting the effect might plateau rather than compound indefinitely on very long, Mars-length missions. That's meaningfully good news for anyone planning a multi-year mission, because it suggests the brain, once it finishes its initial structural adjustment to microgravity, might essentially stabilize in its new configuration rather than continuing to deteriorate for the full duration of the trip. The less reassuring part of the same study is the recovery timeline. It took roughly 3 years for these ventricular changes to fully reverse once astronauts returned to Earth's gravity. 3 years. That means an astronaut who flew a standard 6-month ISS mission may still have had a measurably altered brain structure well over 1,000 days after touching back down on solid ground. This same study turned up another finding worth sitting with. Among astronauts who had flown to space before, ventricles expanded again during a new mission if their previous flight had happened 3 or more years earlier, but they did not expand again if the astronaut had flown within the past 1 to 3 years. Researchers haven't nailed down exactly why, but one plausible reading is that the brain may need a substantial recovery window, measured in years rather than months, before it's fully prepared to safely undergo that same fluid shift stress a second time. It's the kind of finding that raises real, practical questions for how frequently we should be sending the same astronauts back into long-duration missions, questions the field is still actively working through. Think about what that finding actually implies for career astronauts specifically. The small pool of highly trained, experienced professionals space agencies naturally want to keep flying again and again, precisely because of how valuable their accumulated experience is. If a brain genuinely does need something like a 3-year window to fully reset after a 6-month mission before it's ready to safely absorb that same fluid shift stress again, that creates a real, practical tension for crew scheduling. Space agencies want to fly their most experienced people as often as reasonably possible, but this research suggests doing so too soon after a previous long mission might mean sending someone back up before their brain has actually finished settling from the last trip. It's exactly the kind of finding that looks like a narrow technical detail buried inside an MRI study, but actually has direct, practical consequences for how NASA and other space agencies plan astronaut rotations and mission assignments years into the future. One more piece of this puzzle worth mentioning is how researchers managed to study fluid shift effects at all without launching every test subject into orbit. Scientists have found they can partially reproduce the same fluid redistribution seen in real astronauts using something called head-down bed rest studies, in which healthy volunteers here on Earth are kept tilted at a slight head-down angle, sometimes for weeks at a stretch, deliberately encouraging fluid to pool toward the head the same way it does in microgravity. It's a clever, accessible stand-in that lets researchers study some of these mechanisms in far greater numbers and far more cheaply than actual spaceflight would ever allow. And it's produced findings that line up closely with what's been observed in real astronaut brain scans, lending real credibility to the fluid shift explanation as at least part of the underlying cause. It's worth taking a moment to explain what those specific white matter tracts mentioned earlier actually do, because the names alone, corpus callosum, arcuate fasciculus, corticospinal tract, don't mean much without context, and knowing their function helps explain why researchers pay such close attention to changes there specifically. The corpus callosum is the thick band of nerve fibers connecting the brain's left and right hemispheres, essentially the main communication highway, allowing the two halves of the brain to coordinate with one another. The corticospinal tract carries motor commands down from the brain into the spinal cord, forming the core pathway your brain uses to actually move your body on purpose. The cerebellar tracts connect to the cerebellum, the structure primarily responsible for balance, coordination, and fine motor control, which makes it an especially interesting structure to watch for change, given that astronauts are already dealing with a completely disrupted sense of balance and body orientation in microgravity to begin with. Researchers specifically found that spaceflight mission duration was associated with the degree of change in cerebellar white matter, meaning the longer someone stayed in space, the more pronounced this particular structural shift tended to be, a detail that lines up logically with a structure so directly tied to physical balance and coordination being reshaped by an environment where normal balance simply doesn't exist. None of this means these tracts are being damaged in the same destructive sense as the radiation-exposed rodent neurons discussed earlier. Much of this white matter change is better understood as the brain's wiring physically adjusting to a new operating environment, rather than a straightforward injury, but it's still a real, measurable, structural change to some of the most important communication pathways inside the human brain, happening after a period of months spent in an environment nothing in human evolutionary history ever prepared the brain to handle. A closely related 2023 study, again working with a group of 30 astronauts, zeroed in specifically on the lateral and third ventricles, and found that longer missions correlated with greater expansion in those two structures specifically, with the bulk of that expansion happening in the first 6 months and then tapering off on longer missions, echoing the plateau pattern from the study above. This same research team also noticed something related to spaceflight experience itself. Astronauts who had completed more previous missions showed decreases in extracellular free water in the anterior, medial portion of the brain, while less experienced, first-time astronauts showed increases in that exact same brain region. It's a genuinely interesting hint that a brain's response to microgravity might partly depend on whether it's the first time that particular brain has had to make this adjustment, though the underlying biological explanation for that difference isn't fully settled yet either. For context on just how duration-dependent all of this appears to be, one analysis comparing 18 astronauts found that long-duration spaceflight was associated with an average increase in total ventricular volume of about 11%, plus or minus roughly 6%, while short-duration spaceflight showed a change of essentially zero, right around 0.04%. That's not a subtle difference. That's the clearest possible demonstration that time spent in microgravity, not spaceflight itself in some general sense, but specifically the cumulative duration of exposure, is the variable driving these structural brain changes. NASA has been running its own dedicated investigation into exactly this territory, called neuromapping, and an August 2023 update from the agency's Science in Space series described findings tying gray and white matter changes directly to a shift in the brain's internal spatial reference system while in microgravity, specifically affecting tasks that require coordinating sensory input with motor output, things like reaching for an object or navigating your body through a module. NASA's own framing of this research leans notably optimistic, describing it as evidence of the brain's underlying plasticity, its capacity to rewire itself in response to new stimuli, and pointing toward possible future applications in rehabilitating brain injuries and diseases here on Earth. The European Space Agency is running a parallel effort called Brain DTI, specifically investigating whether astronaut brains adapt to sustained weightlessness by strengthening previously underused neural connections, a more targeted look at the same broad neuroplasticity question. And both of these sit inside an even larger 14-study NASA research program called CIPHER, explicitly designed to gather data across missions of varying lengths, so scientists can eventually extrapolate what a genuine multi-year Mars mission might do to the human brain, using the accumulated evidence from years of shorter ISS missions as the foundation. That mention of neuroplasticity is worth unpacking a little further, because it's genuinely one of the more hopeful concepts running through this entire body of research. Neuroplasticity simply refers to the brain's built-in capacity to physically reorganize itself, strengthening some connections, pruning back others, in response to new demands placed on it. The same basic mechanism that allows a person recovering from a stroke to sometimes regain lost function as healthy brain tissue gradually takes over responsibilities from damaged areas, or that allows someone learning a genuinely new skill to develop noticeably faster, more efficient neural pathways for that specific task over time. Framing at least some of what happens to an astronaut's brain in microgravity as neuroplasticity, rather than simple damage, matters because it suggests the brain isn't just passively absorbing punishment from an unfamiliar environment. It's actively working to adapt to it, reorganizing itself to function as well as it possibly can, under conditions nothing in human evolutionary history ever prepared it for. That's precisely why NASA and ESA researchers see such promising potential spillover value for medicine here on Earth. If scientists can better understand exactly how and why the astronaut brain reorganizes itself so effectively in response to a completely novel physical environment, that same understanding could plausibly inform new approaches to rehabilitating people recovering from brain injuries, strokes, or degenerative neurological conditions back on the ground, turning what starts as a space health study into something with a much broader potential medical payoff. A comprehensive review published in The Lancet Neurology in June of 2024 pulled together roughly 15 years of accumulated findings on this entire topic into one summary, and its core conclusion lines up cleanly with everything covered so far. Microgravity produces headward fluid shifts, ventricular expansion, an upward displacement of the brain within the skull, and measurable remodeling of both gray and white matter. The same review also highlighted something worth mentioning on its own, because it connects directly back to that basic problem of not knowing where your own body is in space. Microgravity alters how the brain's vestibular system, the part responsible for balance and detecting head position, processes information, while simultaneously reducing the normal tactile and proprioceptive input, meaning the body's basic sense of its own position and movement that astronauts would ordinarily rely on here on Earth. Put simply, in microgravity, several of the sensory systems the brain has always used to understand where the body is and which way is up either go quiet or start sending confusing signals, and the brain has to find some new way to make sense of the world without them. All of these structural changes converge on one specific, well-documented medical condition that deserves its own dedicated section, because it's arguably the most immediately consequential brain-related effect astronauts experience today, right now, not in some hypothetical future Mars mission, but on current, real ISS missions happening on a regular basis. It's called Spaceflight-Associated Neuroocular Syndrome, or SANS for short, and it sits right at the intersection of the eyes and the brain, which makes sense once you understand that the optic nerve is, in a very real anatomical sense, an extension of brain tissue itself. During early embryonic development, the optic nerve and retina actually form directly out of the same tissue that goes on to become the rest of the central nervous system, meaning that from a strictly anatomical standpoint, looking into someone's eye with the right instrument genuinely does give a doctor a direct, physical window into brain tissue, not just a metaphorical one. That's part of why SANS belongs in a conversation about the brain at all, rather than sitting off to the side as a purely separate eye condition. Whatever is happening to the rest of the astronaut's brain during a long mission is, in a very real sense, also happening to this specific, exposed extension of it. And because the eye is so much easier to directly examine and image than tissue buried deep inside the skull, SANS has become one of the clearest, most accessible windows researchers currently have into how spaceflight is affecting the central nervous system as a whole. SANS is a defined medical syndrome with a specific, recognizable set of features. These include swelling of the optic disc, the point where the optic nerve connects to the retina, a condition called optic disc edema, flattening of the back of the eyeball itself, distension of the sheath surrounding the optic nerve, small cotton wool-like spots appearing on the retina, folds forming in the layer of tissue called the choroid, and a hyperopic shift, meaning astronauts' vision actually becomes measurably more farsighted than it was before the mission. How common is SANS among astronauts on long missions? The honest answer is that published estimates vary somewhat depending on exactly how researchers define and measure it, and presenting that range honestly matters more than picking whichever single number sounds most dramatic. An earlier study by researcher Thomas Mader and colleagues estimated roughly 23% of astronauts on short-duration missions and roughly 48% on long-duration missions reported some degree of visual change. A more recent estimate, from research led by Stephen Laurie, put the incidence among long-duration astronauts specifically at around 70%. Other published sources describe a range closer to 40 to 60% of NASA astronauts on long-duration ISS missions. Different studies, different methodologies, somewhat different numbers, but every single one of them lands on the same underlying conclusion. This is not some rare edge case condition affecting one unlucky crew member out of 100. This is common, arguably closer to the norm than the exception, for anyone who spends an extended period living and working aboard the International Space Station. It's worth pausing on why these prevalence numbers differ from study to study in the first place, because the discrepancy isn't a sign of sloppy science. It's actually a fairly normal feature of how medical research on an emerging condition tends to unfold. Earlier studies, like Mader's, were working with smaller numbers of astronauts and used somewhat looser diagnostic criteria simply because SANS, as a formally named, clearly defined syndrome, hadn't yet been fully standardized at the time that research was conducted. As imaging technology improved, and as more astronauts flew longer missions, giving researchers a larger and more consistent dataset to work from, later studies like Laurie's were able to apply tighter diagnostic definitions and catch more subtle cases that earlier screening methods might have simply missed. That's a pattern worth recognizing anytime you see prevalence numbers climb over time in a relatively young area of medical research. It doesn't necessarily mean the underlying condition is becoming more common. It often just means researchers are getting better at actually detecting it. What actually causes SANS remains genuinely unresolved, and researchers studying it are refreshingly upfront about that uncertainty, rather than pretending the mechanism is fully understood. The leading hypotheses currently under investigation include the same headward fluid shift already discussed at length, possible mild and transient elevations in intracranial pressure, elevated carbon dioxide levels inside the spacecraft cabin itself, individual genetic predisposition that might make some astronauts more susceptible than others, and impairment of that glymphatic waste clearance system mentioned earlier. Some features of SANS overlap diagnostically with a separate, Earth-based condition called secondary pseudotumor cerebri syndrome, though researchers are careful to note the two aren't identical, and exactly how they relate to one another is still being worked out. One particularly telling clinical detail underscores just how much remains unknown. In some documented cases, the optic nerve swelling associated with SANS has persisted for up to 6 months after an astronaut's return to Earth, even in cases where their intracranial pressure had already measured back to normal, which strongly suggests intracranial pressure alone can't be the whole explanation, whatever the full story eventually turns out to be. The fact that researchers are actively pursuing 5 or more distinct hypotheses simultaneously, rather than converging on one dominant explanation, tells you something honest about where this specific area of research currently stands. It's genuinely possible, and increasingly looks likely based on the accumulated evidence so far, that SANS isn't caused by any single mechanism acting alone, but by some combination of several of these factors interacting together: the fluid shift altering pressure dynamics, elevated cabin carbon dioxide compounding that effect, individual genetic differences determining how much any given astronaut's eyes and brain can tolerate before symptoms actually appear, and a possibly already compromised glymphatic drainage system struggling to keep up with all of it at once. That kind of multi-factor explanation is common in medicine generally, particularly for newer, still developing syndromes. And it's part of why researchers studying SANS have been careful not to prematurely settle on one tidy causal story before the evidence genuinely supports it. Getting the explanation wrong, or oversimplified, carries real consequences too, since any protective countermeasure eventually developed for SANS will only work if it's actually targeting the true underlying cause, or causes, rather than a plausible-sounding theory that turns out to only be part of the real picture. Treating SANS in orbit presents its own unique set of headaches, quite apart from simply understanding what causes it. Standard treatments for eye conditions on Earth typically rely on things like eye drops, administered on a regular schedule, which is a genuinely awkward proposition in microgravity, where a stray drop doesn't simply roll off the eye and down the cheek the way it would under normal gravity, but instead floats, breaks into smaller droplets, and can drift into places it was never meant to go. That's a small, almost mundane detail, but it's a good example of how a lot of terrestrial medicine simply doesn't translate cleanly into a weightless environment, and it's part of why researchers are so focused on prevention and root cause understanding rather than relying on treatment after the fact. Because of how directly relevant this is to any future Mars mission, where evacuation to a hospital on Earth simply isn't an option, SANS ranks among the most actively studied astronaut health risks in the entire field right now, with newly published expert consensus papers still working, as of the most recent research available, to standardize exactly how it should be diagnosed and managed going forward. Now let's shift away from the brain's physical structure for a moment and talk about something a little more familiar to anyone who has ever pulled an all-nighter or struggled through a rough week on too little rest, because it turns out astronauts deal with a version of that same struggle almost constantly, and the data on exactly how much sleep they're actually getting up there is more striking than you might expect. Sleep deprivation and circadian rhythm disruption are described in the scientific literature as highly prevalent among astronauts, in some ways comparable to what shift workers experience here on Earth, except with the added complications of living in an unfamiliar 16 sunrises a day environment, cramped quarters, constant ambient noise from life support systems, and the psychological weight of the mission itself. One large review pulled together sleep data from actual ISS crew members and found that during spaceflight, astronauts averaged just 6.09 hours of sleep per night, plus or minus about 40 minutes, compared to 6.95 hours per night after they'd returned to Earth, a statistically significant gap of nearly an hour. Even more striking, that same dataset found astronauts failed to get adequate sleep on 47.1% of the nights studied, 480 out of 1,020 total nights recorded. That's not an occasional rough night here and there. That's essentially a coin flip, night after night, for the entire duration of a mission. Given how common this is, it probably won't surprise you that sleep-promoting medication use is widespread among astronauts, common enough that researchers studying the issue flag it as a genuine concern in its own right, particularly given the detail that some crew members reportedly take a second dose of medication partway through the night, which researchers interpret as a sign the first dose may not have been fully effective. That pattern of unrestricted, repeated medication use is something researchers explicitly describe as a concern worth addressing directly, not simply a minor footnote, given how heavily astronauts are already relying on pharmaceutical support just to get through a normal night's sleep in orbit, on top of everything else their bodies are already managing. NASA takes astronaut cognitive functioning seriously enough that it's built directly into the mission's standard operating procedure. The agency uses a specific tool called the Psychomotor Vigilance Task, often abbreviated PVT, as part of what's officially called Standard Measures, a routine, ongoing part of how NASA actively monitors crew cognitive performance throughout an entire mission, not just before or after it. The test itself is deliberately simple by design, typically asking a crew member to respond as quickly as possible to a visual stimulus appearing on a screen at unpredictable intervals, with both reaction speed and the number of missed responses, called lapses, recorded and tracked over time. Its simplicity is actually the point. A test that takes only a few minutes and requires no special training can be repeated frequently enough, day after day, throughout an entire mission, to build a genuinely useful longitudinal picture of how alert and responsive each individual astronaut is on any given day, functioning almost like a daily vital sign for cognitive readiness, rather than a one-time snapshot. One study found astronauts who got 5 hours of sleep or less on a given night aboard the ISS performed measurably worse the next day, specifically on tasks requiring sustained attention. Exactly the kind of finding you'd expect, based on decades of terrestrial sleep research. But here's where the space-specific data gets genuinely more nuanced than you might assume. A large cognitive performance study looking at ISS astronauts across full 6-month low Earth orbit missions found no evidence of a systematic, overall decline in cognitive performance across the entire mission. That's a meaningfully reassuring headline finding on its own. But dig one layer deeper into the same study, and the picture gets more textured. Researchers did find slowed performance, specifically early in the mission, on tasks measuring processing speed, visual working memory, and sustained attention, along with a documented decrease in risk-taking propensity during the later part of the mission and immediately after landing, which makes a certain intuitive sense. An astronaut approaching a high-stakes landing procedure might reasonably become more cautious rather than less. Perhaps most interesting of all, when researchers looked beyond simple group averages and instead tracked how far individual astronauts' scores had shifted from their own personal pre-flight baseline, they found that 11.8% of all in-flight and post-flight cognitive test scores across the entire study fell at or below 1.5 standard deviations under that individual's own baseline. Nearly 1 in 8 test results represented a meaningful dip below where that specific astronaut normally performed, even though the group's overall average looked essentially stable. Group averages, in other words, can hide real individual struggles happening underneath them. It's worth pausing on that early flight slowdown specifically, because it lines up neatly with everything else this video has already covered about what's physically happening to an astronaut's body and brain in those first days and weeks after launch. That's precisely the window when the cephalad fluid shift is at its most dramatic and least adapted to, when the vestibular system is still recalibrating to a complete absence of a reliable down, and when the body is generally working hardest just to figure out how to function in an entirely new physical environment. It would actually be more surprising if processing speed and working memory didn't take a temporary hit during that specific adjustment window, given how much else is going on inside the astronaut's nervous system at exactly the same time. The fact that this early dip shows up consistently and then appears to resolve as the mission continues fits a coherent story of adaptation rather than ongoing decline. One more small piece of evidence supporting the broader theme running through nearly all of this structural brain research. A lot of what looks alarming on first glance turns out to be the brain going through a genuine adjustment period, not a one-way slide toward permanent impairment. That distinction between a genuine adjustment period and permanent ongoing decline is worth carrying with you as the single most important thread running through this entire video, because it's easy to hear terms like chromosomal rearrangement, ventricular expansion, and dendritic damage stacked one after another and walk away with an impression far more alarming than the underlying research actually supports. The honest picture is more layered than that. Some of what deep space does to the brain genuinely does look like damage, and researchers describe it in exactly those terms, particularly in the cosmic radiation research. Some of it looks more like adaptation, the brain actively reorganizing itself to function in an environment nothing in human evolution ever prepared it for. And some of it, the overview effect specifically, looks like something closer to expansion, a genuinely new kind of human experience, made possible only by leaving the planet in the first place. Keeping those three categories distinct, rather than collapsing everything into one vague, ominous idea that space simply breaks the human brain, is really the entire point of walking through this evidence as carefully and specifically as this video has tried to do. One more finding from this same body of research is worth calling out specifically because it cuts against a natural assumption. You'd probably expect a tight, reliable relationship between how much sleep an astronaut reports getting and how well they perform cognitively the next day. This particular large-scale study found no clear, consistent statistical link between the two, a result the researchers themselves acknowledged as somewhat surprising given what's generally understood from sleep research conducted on Earth. Other individual studies referenced within that same broader review documented additional specific effects worth noting. Slowed reaction time on spatial working memory tasks, measured after return to Earth rather than during the mission itself. Noticeable decrements on visuospatial line orientation tasks, both while still in flight and shortly after landing, and a documented increase in errors on a dual tasking, essentially a multitasking test, specifically on the actual day of landing, arguably one of the worst possible moments for an astronaut's cognitive performance to dip, given how demanding a real landing procedure is. The genuinely good news buried in all of this is that in every one of these specific cases, the post-flight cognitive decrements researchers documented returned to each astronaut's pre-flight baseline within about 30 days of touching back down on Earth. Ground-based research offers one more relevant data point here, and it connects sleep loss directly to one of the most operationally critical tasks an astronaut can be asked to perform. Researchers studying the effects of a single night of total sleep deprivation on a simulated manual spacecraft docking task found measurable impairment in docking performance, alongside the expected decline on the standard attention-based PVT test. Manual docking is a real skill astronauts sometimes have to execute under genuine pressure, and this research underscores that sleep loss doesn't just make someone feel foggy or tired. It measurably degrades performance on exactly the kind of high-stakes technical task where degraded performance actually matters. None of this has gone unnoticed by mission planners, who have spent years developing countermeasures aimed specifically at protecting astronauts' sleep and alertness. NASA has experimented with specialized lighting systems, sometimes called solid-state light assemblies, designed to help regulate crew melatonin production and keep circadian rhythms better aligned despite the complete absence of a natural day-night cycle aboard a spacecraft orbiting the planet every 90 minutes. Napping has emerged as another common, informal strategy, with ISS crew members frequently using scheduled daytime naps to help offset accumulated sleep debt, though researchers note that poorly timed naps can sometimes backfire, disrupting circadian rhythm further and producing grogginess rather than relief. Some astronauts have also reported using stimulants like caffeine or the prescription alertness medication modafinil to push through periods of insufficient sleep, though researchers tracking this have found that astronauts who rely on these stimulants also tend to report more difficulty falling asleep afterward, suggesting a cycle where the fix for one night's alertness problem can end up feeding the next night's sleep problem. This isn't a new concern either. Fatigue linked to circadian desynchronization and sleep loss showed up as far back as the Gemini and early Skylab era missions, decades before the ISS existed, meaning sleep disruption has been a documented feature of human spaceflight for essentially the entire history of the space program, not some new problem unique to long-duration missions. More recent reviews of astronaut sleep research have expanded the list of countermeasures under active investigation well beyond lighting and medication. Researchers have started exploring whether modulating an astronaut's gut microbiota, the community of bacteria living in the digestive system, might help support healthier sleep and circadian regulation during long missions, based on growing evidence from Earth-based research linking gut health to sleep quality more broadly. Some reviews have even discussed approaches drawn from traditional Chinese medicine as a potential complementary avenue worth investigating further, alongside more conventional pharmacological and lighting-based interventions, reflecting just how broad a net researchers are currently casting in the effort to solve astronaut sleep problems from as many angles as possible. None of these newer approaches have replaced the established tools already in use, the specialized lighting, the carefully scheduled naps, the standard sleep medications, but their inclusion in current research reviews signals that mission planners aren't treating astronaut sleep as a solved problem, even after decades of accumulated data on exactly how disruptive spaceflight tends to be to a normal night's rest. If anything, the growing list of countermeasures under active study reflects a field that increasingly treats sleep loss not as a minor inconvenience astronauts simply have to push through, but as a genuine operational risk worth solving with the same seriousness applied to radiation shielding or structural brain change. That kind of parity in how seriously mission planners now treat sleep alongside radiation and structural brain change represents a real shift in perspective from earlier decades of spaceflight, when tired astronauts pushing through a demanding schedule was sometimes treated as simply part of the job rather than a genuine, measurable risk to mission safety and crew cognitive performance worth actively engineering around. So far, everything covered in this video has been about the brain in a fairly literal, physical sense. Chromosomes, neurons, fluid shifts, ventricles, optic nerves, sleep architecture. But there's an entirely different category of evidence worth walking through now, because it doesn't come from MRI machines or blood samples at all. It comes from watching what happens to the human mind, not the tissue, but the actual lived psychological experience, when you take a small group of people and seal them away from the rest of the world for months or years at a time. And for this line of research, scientists haven't needed to wait for an actual Mars mission to launch. They've been running remarkably faithful ground-based simulations for well over a decade. The most ambitious of these is a project called Mars 500, led by the State Scientific Center of the Russian Federation's Institute of Biomedical Problems in Moscow. Mars 500 wasn't a single experiment. It unfolded across 3 distinct phases: a short 14-day pilot study completed in November of 2007, a longer 105-day pilot completed in July of 2009, and finally, the main event, a full 520-day isolation study that concluded on November 4, 2011. 520 days, nearly a year and a half, sealed inside a mock spacecraft facility, chosen specifically because it closely approximates the actual estimated duration of a real Mars mission. The 520-day crew consisted of 6 men, 3 Russians, 1 Frenchman, 1 Italian-Colombian, and 1 Chinese crew member, with an average age of about 32.4 years. Every detail of the simulation was engineered for realism, right down to the communication delay. Messages between the crew and mission control outside were deliberately delayed by an average of 13 minutes each way, faithfully reproducing the actual signal lag a real Mars-bound crew would experience, meaning something as simple as asking a question and getting a response could take well over 25 minutes round trip. No live conversation, no quick reassurance in a crisis, just a slow, deliberate exchange of messages across a simulated gulf of tens of millions of miles. The crew even carried out 3 simulated Mars surface walks on February 14th, 18th, and 22nd of 2011, stepping outside their mock spacecraft in full simulated extravehicular gear as though they'd actually landed. The physical facility itself was purpose-built for this exact kind of long-term psychological research, located at the Institute of Biomedical Problems site in Moscow, and consisting of a sealed isolation module, a separate mission operations room where outside researchers monitored the crew, technical support facilities, and offices, all designed to function as a self-contained little world the crew would not leave, in any capacity, for the entire 520 days. Putting together a crew that international wasn't an incidental detail either. Real Mars missions, if and when they happen, will almost certainly involve multinational crews, given how expensive and technically demanding deep space travel is, meaning any useful isolation research needs to account for how people from meaningfully different cultural and linguistic backgrounds actually get along, communicate, and resolve conflict when there's genuinely nowhere else to go and no one else to talk to. 3 Russians, a Frenchman, an Italian-Colombian, and a Chinese crew member, spending 520 consecutive days together, sharing meals, resolving disagreements, and managing the inevitable friction of a small group living in close quarters, produced exactly the kind of cross-cultural interpersonal data a purely single-nationality crew simply couldn't have generated. The official summary of how Mars 500 concluded is notably upbeat. Organizers described the crew as ending the experiment in what they characterized as optimal physical and psychological condition. And on the surface, that's a genuinely encouraging result. 6 people spending nearly a year and a half together in extreme confinement and coming out the other side reportedly fine. But peer-reviewed follow-up research told a somewhat more complicated story. A report published in the Proceedings of the National Academy of Sciences in February of 2013 found that 4 of the 6 crew members developed considerable sleep problems during the mission, along with increased overall sleep and rest time, in a pattern researchers described as behaviorally resembling animal hibernation, alongside broader disruption to their normal circadian rhythm throughout the confinement period. That's a striking contrast worth sitting with for a moment. The official mission summary said optimal condition. The peer-reviewed academic follow-up found 2/3 of the crew developing real, measurable sleep and rhythm disorders. Both accounts are legitimate and both come from credible sources. It's simply a reminder that official mission wrap-ups and detailed academic publication sometimes end up emphasizing rather different pieces of the same underlying story. Throughout the mission, researchers tracked the crew's psychological state using an established battery of tools, administered weekly: the Beck Depression Inventory, the Profile of Mood States Questionnaire, a dedicated conflict questionnaire, the same psychomotor vigilance test used aboard the actual ISS, and a series of visual analog scales measuring stress and fatigue. What emerged from all this careful tracking was substantial variation between individual crew members in how they psychologically handled the same shared confinement, some adapting relatively smoothly, others clearly struggling more, a pattern that echoes almost every other isolation study ever conducted and reinforces just how individually variable this kind of psychological stress really is. A separate, more specific psychological analysis of the Mars 500 data used something called the International Affective Pictures System, a standardized set of images designed to evoke measurable emotional responses, to track how the crew's emotional processing shifted over the course of the full 520 days. The finding was subtle but genuinely fascinating. As confinement dragged on, crew members increasingly rated negative, unpleasant images more positively than they had earlier in the mission. Researchers interpreted this as a possible unconscious defensive psychological adaptation, essentially the mind gradually softening its own emotional response to negativity as a coping mechanism for an unrelentingly monotonous and stressful environment it couldn't simply walk away from. This shift followed what researchers described as a distinct, staged pattern, one that echoed a phenomenon already well documented in other isolation research, sometimes called the third quarter phenomenon, referring to a well-established dip in morale that tends to show up roughly 3/4 of the way through a long isolation period, right when the initial adrenaline of the mission has worn off, but the eventual relief of returning home still feels impossibly far away. Despite all of this, crew log entries from the mission described high enthusiasm and strong motivation throughout, which the crew themselves credited as a major factor helping them manage anxiety and adapt successfully to their unusual and unchanging surroundings. That third quarter phenomenon is worth dwelling on for a moment longer, because it turns out to be one of the more reliable, predictable patterns in the entire isolation research literature, showing up not just in Mars 500, but across decades of Antarctic and polar research going back long before anyone was simulating a Mars mission specifically. The basic shape of it is intuitive once you hear it described. Early in any long isolation period, there's typically a burst of novelty and purpose. Everyone is focused, motivated, and still running partly on the adrenaline of having just begun something genuinely difficult and meaningful. Somewhere around the halfway point, that initial energy tends to fade, replaced by the grinding reality of routine. But it's specifically that third quarter stretch, not the middle, but later, when the finish line has become theoretically visible but still remains agonizingly far off, where morale tends to hit its lowest point. One researcher writing on Mars 500 crew's own log entries directly tied this pattern to the crew's own written record, noting that the crew's log, kept by one of the crew members who also happened to be the study's first author, showed exactly this kind of documented emotional dip appearing on schedule. Understanding this pattern isn't just an academic curiosity either, it's directly useful for real mission planning. Because if psychologists and mission support teams know in advance roughly when a crew is statistically most likely to struggle, they can proactively schedule additional support, communication, or morale-focused interventions to land exactly during that vulnerable window, rather than only reacting after problems have already surfaced. Mars 500 isn't the only major isolation analog worth mentioning here. NASA has funded its own series of Mars analog missions called High Seas, short for the Hawaii Space Exploration Analog and Simulation, where small crews live inside a geodesic dome perched on an isolated volcanic site on Hawaii's Mauna Loa, cut off from normal daily contact with the outside world, for mission lengths ranging anywhere from 4 to 12 months, depending on the specific expedition. One particular high seas mission followed 6 crew members, 3 men and 3 women, through a full 8-month isolation period, specifically testing a computer-based mental health support tool called the Virtual Space Station, built around interactive modules addressing conflict, stress, and depression. Chosen specifically because a computer-based tool offers confidentiality and doesn't require reaching out to anyone beyond the isolated crew itself. Researchers reported that this crew experienced the kind of behavioral health challenges commonly seen across isolated, confined environments generally, and found the support tool genuinely valuable, both used exactly as intended and, interestingly, in some unanticipated ways the researchers hadn't originally planned for. Researchers studying HIC's crews more broadly, across the program's various expeditions, have documented the same general emotional arc seen in Mars 500, declines in mood over time, increased interpersonal tension, and reduced motivation as a mission wears on, with crew cohesion fluctuating rather than holding steady, and periods of heightened conflict tending to cluster specifically around the middle portion of each expedition. Some more recent research has even tried to model this mathematically, using what's called agent-based modeling, essentially building computer simulations of hypothetical Mars crews using established psychology frameworks like the 5-factor model of personality, which measures traits including openness, conscientiousness, extroversion, agreeableness, and neuroticism, to predict how different combinations of crew personalities and skills might hold up or fall apart over a full 500-day simulated mission. The goal of that kind of modeling is directly practical. If mission planners can get a reasonable predictive sense of which personality combinations tend to produce more resilient, better functioning crews before anyone actually launches, that becomes a genuinely useful tool for astronaut selection and crew assignment on a real, multi-year Mars mission, where getting crew dynamics wrong isn't something you can simply fix by sending in a replacement partway through. Zooming out even further, an entire broader category of research exists around what scientists call ICE environments, short for isolated, confined, and extreme, encompassing not just Mars analogues like Mars 500 and high seas, but also genuine Antarctic research stations, including Concordia, Halley 6, Rothera, and Neumeier 2, along with various polar expeditions. Across this entire body of research, spanning decades and multiple continents and dozens of different crews, a remarkably consistent pattern shows up again and again. Mood tends to decline over the course of long confinement. Interpersonal tension tends to increase. Motivation tends to decrease. Crew cohesion fluctuates rather than staying stable. And conflict among crew members tends to peak specifically during the middle portion of a mission, a pattern researchers refer to simply as the mid-mission phase. Researchers studying these Antarctic and polar analog environments have gone further than simply cataloging mood swings too. Some of this work involves directly interviewing people who have actually lived through these deployments, gathering first-hand accounts from sojourners who spent time at stations like Concordia, Halley 6, Rothera, and Neumeier 2, or on extended polar treks, specifically to understand the demands of that kind of isolation from the inside, at every level of daily functioning, not just from questionnaires and hormone panels. What emerges from decades of this combined research is a fairly consistent human portrait, regardless of whether the isolation happens in a mock spacecraft in Moscow, a dome on a Hawaiian volcano, or a research station buried in Antarctic ice. The specific setting changes. The underlying psychological pattern really doesn't. People placed in prolonged confinement, cut off from their normal social world, tend to move through recognizable emotional stages, an initial period of high motivation and novelty, a middle stretch where friction and fatigue tend to build, and eventually some form of adjustment or accommodation as the group settles into whatever version of normal is available to them within those walls. That consistency across such wildly different physical environments is itself a meaningful finding. It suggests the psychological toll researchers are measuring isn't really about spacecraft or domes or ice stations specifically. It's about isolation and confinement as a category of human experience, one that produces broadly similar effects on mood and group dynamics, no matter where on Earth, or eventually off it, you happen to be experiencing it. That's a genuinely useful conclusion for mission planners specifically, because it means decades of pre-existing Antarctic and polar research gathered long before anyone was seriously planning a Mars mission, still carries real, transferable scientific value today. Space agencies don't need to start entirely from scratch every time they want to understand how a crew might psychologically hold up over a multi-year mission. They can draw on an already substantial body of evidence from Antarctic winter rover crews who have been living through months of total isolation, extreme cold, and complete darkness for decades, long before Mars 500 or HICs existed at all. That overlap between space psychology and polar psychology has become its own small but genuinely productive area of research collaboration, with researchers from both fields regularly citing each other's work, comparing notes across what look like completely different settings on the surface, a spacecraft mock-up and an ice station separated by thousands of miles, but which turn out to be remarkably similar from the specific standpoint of what isolation and confinement actually do to the human mind over time. Here's why all of this matters so directly for a real future Mars mission, beyond simply being an interesting body of psychological research. Using current propulsion technology, a genuine crewed Mars mission is estimated to require somewhere around 2.5 to 3 full years, once you account for the outbound transit, meaningful time spent on the Martian surface conducting the actual mission, and the return journey home. That's noticeably longer than even the record-setting 520-day Mars 500 simulation, meaning whatever psychological strain researchers have already documented in these ground-based analogs would need to be sustained even longer on an actual mission, and every single researcher working in this space is careful to flag an important limitation of their own findings. However rigorously designed these ground simulations are, they cannot fully replicate the psychological reality of an actual space mission, specifically because simulation crews always know, somewhere in the back of their minds, that rescue or early departure remains technically possible if something goes seriously wrong. A real Mars crew wouldn't have that same safety net, and stacked on top of that missing safety net would be the added, genuine physiological stress of true microgravity and real cosmic radiation exposure, layered directly on top of the isolation itself, rather than isolation being studied in relative comfort back here on Earth. That layering effect is really the key idea to hold onto from this entire section, and it connects directly back to everything covered earlier in this video. Every psychological finding from Mars 500 and HICs, the mood decline, the third quarter dip, the increased interpersonal friction, was measured in volunteers who were otherwise healthy, well-fed, breathing normal air, sleeping in normal gravity, and facing no actual cosmic radiation exposure whatsoever. A real Mars crew would be experiencing all of that same psychological strain simultaneously with everything else this video has already covered: the chromosomal changes, the shrinking dendrites, the expanding ventricles, the chronic sleep debt, and the SANS-related visual changes, all stacking on top of each other for 2.5 to 3 years straight, with no early exit available if the combined weight of it all became too much for any individual crew member to bear. None of the individual ground-based studies covered in this video were ever designed to capture that full, compounded picture on their own. And that's precisely why mission planners treat the psychological research, the structural brain research, and the radiation research as 3 separate but tightly related pieces of the same overall risk assessment, rather than 3 unrelated fields that happen to all involve astronauts. Before moving into the last major piece of this story, it's worth pausing to lay out how all of these individual findings actually line up against each other in time, because seeing the chronology helps clarify just how recent and how rapidly accelerating this entire field of research really is. The groundwork stretches back further than you might expect. The 3 Mars 500 pilot and main studies ran between November of 2007 and November of 2011, with the follow-up PNAS paper on crew sleep disruption appearing in February of 2013. Around that same period, in 2013, the earlier mouse study linking radiation exposure to Alzheimer's-like changes was published, planting an early seed for what would become Limoli's more extensive work. Scott Kelly launched to the ISS in March of 2015, beginning the 340-day mission at the heart of the twins' study, with his brother Mark serving as the ground-based control for the full duration. Limoli's team published their major cosmic radiation and brain damage findings in October of 2016, The same month, a sitting American president publicly announced ambitions to send astronauts to Mars within roughly the following 2 decades. A genuinely striking coincidence of timing between political ambition and sobering biological evidence. A separate study connecting Apollo-era radiation exposure to cardiovascular damage also emerged that same year. The full twin study results were finally published in Science in April of 2019. The same year, Limoli's team published their chronic, low-dose follow-up radiation study in the journal eNeuro, and the same year a detailed white matter and free water diffusion MRI study using data from 15 astronauts appeared as well. By 2021, both NASA's neuromapping investigation and ESA's brain DTI investigation were actively running aboard the ISS, and by 2022, researchers had published their detailed comparison of perivascular space changes between NASA and Roscosmos crew members. The year 2023 alone produced an unusually dense cluster of major findings. The June Scientific Reports study establishing that ventricular expansion plateaus around the 1-year mark and takes roughly 3 years to fully reverse, a related study on spaceflight experience and free water distribution, and NASA's August Science in Space update detailing the neuromapping and cipher findings on neuroplasticity. In 2024, The Lancet Neurology published its comprehensive review, pulling together roughly 15 years of accumulated brain research into one summary, alongside a further longitudinal perivascular space study. And as of the most current research available heading into 2025 and 2026, work on standardizing SANS diagnosis and management continues, alongside ongoing NASA and ESA in-flight cognitive monitoring, meaning this entire area of science remains very much a live, moving target, rather than a settled, closed body of knowledge. Anyone watching this video today is genuinely watching a research field still very much in progress, with the next major finding likely already sitting somewhere in a lab, a peer review queue, or a fresh batch of returning astronaut MRI scans waiting to be published. It's also worth being upfront about a handful of genuine tensions and open questions running through all of this research, rather than presenting it as one single, unified, uncontroversial conclusion, because the honest picture is more complicated than that. First, NASA's own various summaries of the twins study aren't perfectly consistent with one another, with one account emphasizing a documented cognitive decline in memory and logic testing, and another emphasizing that cognition remained largely stable across most measures. Both come from credible, NASA-linked sources, and the fair reading is that the full cognitive test battery simply produced a genuinely mixed result across different mental domains, not one single clean verdict in either direction. Second, there's a real, unresolved tension between the structural MRI evidence and the cognitive performance evidence more broadly. The MRI data showing ventricular expansion, fluid shifts, and white matter change is remarkably strong and consistent across many independent studies. But the large-scale cognitive performance research, covering typical 6-month ISS missions, found no clear evidence of systematic overall cognitive decline. In plain terms, the brain visibly and measurably changes shape during spaceflight, but scientists have not yet clearly proven that this physical change reliably translates into meaningful, mission-relevant impairment in thinking ability, at least not on missions of the length flown so far. Whether that link becomes clearer or more concerning on a much longer Mars-length mission remains one of the central open questions driving current research. Third, every one of those striking radiation extrapolations, including the estimate that at least one crew member in a 5-person Mars crew might develop severe cognitive deficits, comes explicitly from rodent data scaled up mathematically to project human risk. No human being has ever actually been exposed to that level of deep space cosmic radiation for that length of time, so these figures remain projections built on the best available evidence, not confirmed human outcomes. And fourth, as already mentioned, Mars 500's own official summary describing the crew as ending in optimal condition sits somewhat uneasily next to the independently published, peer-reviewed finding that 4 of the 6 crew members developed real sleep and circadian problems. A useful reminder that official mission narratives and academic publication don't always tell exactly the same story, and that both are worth reading rather than picking just one. There's a fifth tension worth naming too. one that runs underneath essentially everything else in this video, rather than belonging to any single finding. The overview effect is treated with real seriousness by researchers, astronauts, and space agencies alike, and it's backed by decades of remarkably consistent first-hand testimony, plus at least one formal behavioral study tracking real changes in environmental attitude. And yet, unlike the ventricular expansion, the dendritic damage, or the sleep deprivation data, it hasn't been pinned down as a specific, measurable neurological event the way those other findings have. That's not a flaw in the science covered in this video. It's simply an honest acknowledgement that not every real, well-documented human experience related to spaceflight has yet been fully translated into hard neuroscience, and pretending otherwise, in either direction, either dismissing the overview effect because it lacks an MRI signature, or overselling it as fully proven brain science, would misrepresent exactly where the evidence currently stands. After walking through chromosome damage, shrinking dendrites, expanding ventricles, swollen optic nerves, chronic sleep debt, and the psychological grind of isolation, it would be easy to walk away from this video thinking deep space is purely and simply bad news for the human brain. But that would leave out what might be the single most fascinating and most consistently reported psychological effect of spaceflight of all, and it's overwhelmingly positive. It's called the overview effect, and unlike almost everything else covered in this video, it wasn't discovered in a lab. It was named by an author and space philosopher named Frank White back in 1987, in his book, The Overview Effect, Space Exploration and Human Evolution. White developed the concept the old-fashioned way, by directly interviewing astronauts about what it actually felt like to look back and see the entire Earth suspended against the total blackness of space. At the time White coined the term remember, only about 300 human beings total had ever been to space, a strikingly small pool of people who had personally witnessed what he was trying to describe. Academic research building on White's original observation has since characterized the overview effect in fairly precise psychological language, describing it as a state of awe carrying self-transcendent qualities, triggered by an unusually striking, direct visual stimulus, specifically the sight of the whole Earth from a distance. Astronauts who have experienced it consistently report a genuine shift in their own self-concept and value system afterward. A 2020 study by researcher Anais Vosky specifically documented a measurable influence on astronauts' environmental attitudes and behavior following spaceflight, essentially finding that people who have personally seen Earth as a single, fragile, borderless sphere floating in the void tend to come back caring measurably more about protecting it. Former NASA astronaut Leland Melvin has said the effect seems to take hold of astronauts regardless of their culture or country of origin, suggesting something close to a universal human response underneath it. Other researchers studying the phenomenon, including a team led by David Yaden, found that while something like a shared core experience does appear to happen broadly across astronauts, cultural, religious, and social background does noticeably shape how each individual interprets and describes it afterward, ranging anywhere from explicitly religious language to something more vaguely spiritual, to purely naturalistic scientific description, to simply a strong, practical sense of social or environmental responsibility. That academic characterization mentioned earlier, describing the effect as a state of awe carrying self-transcendent qualities, is worth sitting with for a moment, because both of those specific words are doing real work in how psychologists think about this phenomenon. Awe, in the formal psychological sense researchers use the term, generally describes an emotional response triggered by encountering something that feels vast, that doesn't fit neatly into a person's existing mental framework for understanding the world, and that as a result tends to force some degree of genuine reassessment of how that person sees things going forward. Self-transcendence, meanwhile, refers specifically to a temporary or lasting shift in which a person's sense of self expands outward, feeling less like a small, separate individual and more connected to something larger than themselves. Put those two ideas together, and you get a fairly precise psychological description of exactly what astronauts have been describing in their own words for decades, encountering something too vast to easily process, the whole Earth, borderless, fragile, and alone against the black, and coming away from that encounter with a genuinely altered, more expansive sense of their own place within it. Frank White himself has described the effect as fundamentally beyond words, difficult to convey secondhand no matter how carefully you try. At one point comparing that difficulty to trying to explain a genuine Zen Buddhist realization to someone who has never experienced anything like it directly. In his own framing, astronauts come to understand experientially something the rest of us can really only grasp intellectually from down here, that we are, in a very real and literal sense, living on a planet that is itself a kind of spaceship, moving through the universe at enormous speed, with every single one of us effectively serving as crew aboard it. A framing White deliberately borrows from Buckminster Fuller's earlier concept of Spaceship Earth. A documentary film built directly around White's research and interviews, simply titled Overview, has been watched more than 8 million times online, a genuinely massive audience for a film essentially about a psychological phenomenon. It's worth dwelling for a moment on that Vosky research mentioned earlier, because it's one of the few pieces of this puzzle that moves beyond simple testimony and into something closer to measurable, tracked behavior. Rather than just asking astronauts to describe how the experience felt in the moment, that 2020 study specifically examined environmental attitudes and behaviors after the fact, looking for a lasting influence rather than a fleeting emotional reaction during the flight itself. What it documented was a new level of environmental awareness and consciousness among astronauts who had personally seen the planet from that vantage point, a finding that fits neatly with decades of anecdotal astronaut testimony describing exactly that kind of shift, but this time backed by something closer to a formal behavioral study rather than a moving quote in a documentary. White has spoken about wanting to take this idea even further, describing an ambition he calls bringing the overview effect down to Earth, essentially trying to find ways for people who will never personally leave the planet to still internalize some version of that same unified, borderless perspective on our shared situation. His broader argument is that humanity needs to grow into something closer to a genuinely global perspective, a habit of seeing every other person as effectively a fellow crew member on the same shared vessel, before we're truly ready to expand further out into the solar system in any serious, sustained way. Whether or not you find that framing persuasive, it's a genuinely different note to end a story about brain science on, considering everything else covered in this video involves damage, disruption, and things going wrong. The overview effect is the one major finding in this entire body of research that consistently points the other direction entirely, toward something astronauts overwhelmingly describe as expansive, clarifying, and, in the specific language used by researchers studying it, genuinely awe-inducing. It's worth being just as careful and honest about the overview effect as we've tried to be about every other finding in this video, though. In a 2026 interview, White himself drew a clear, deliberate line around exactly how much of this phenomenon has actually been scientifically confirmed. In his own words, the overview effect is real, measurable in its consequences, if not yet in its neurology. That's a genuinely important distinction to sit with. Everything covered earlier in this video, the ventricle expansion, the dendritic damage, the sleep data, the fluid shifts, comes from direct neurological and physiological measurement, MRI scans, blood tests, chromosome analysis, standardized cognitive testing. The overview effect, by contrast, rests almost entirely on decades of consistent, compelling first-hand testimony from astronauts themselves, and on its measurable downstream consequences, like documented shifts in environmental attitude and behavior, rather than on any confirmed, isolated neurological mechanism inside the brain. It is very likely real. It is consistently and independently reported by people with no obvious reason to coordinate their stories. But it hasn't yet been pinned down at the level of neuroscience the way the structural brain changes discussed earlier in this video have been. And that distinction matters if we want to represent the full picture honestly rather than treating every space-related brain claim as equally proven. Looking forward, White has suggested that as commercial spaceflight continues expanding access to space, beyond the small handful of career astronauts who experienced it in the decades since his book was first published, potentially bringing spaceflight within reach of far more people than the original 300 pioneers he interviewed, we may be approaching a moment when meaningfully larger numbers of human beings get to personally experience this particular cognitive shift for themselves, whatever its precise underlying neuroscience ultimately turns out to be. Companies including SpaceX, Blue Origin, and Virgin Galactic have all been working to develop commercial spaceflight capability, part of a broader industry push to move space travel beyond the small, highly selective pool of government-trained career astronauts, who made up essentially the entire population of space travelers for the first several decades of human spaceflight. If that access genuinely continues expanding the way White anticipates, the overview effect may end up being studied not just as a rare psychological phenomenon experienced by a few hundred elite, highly trained professionals, but as something closer to a documentable, repeatable response researchers can study across a much larger and more demographically varied population of people, which would go a long way toward finally answering the exact question White himself has left open: whether this remarkable, consistently reported shift in awareness has a specific, identifiable neurological signature after all, once there are simply enough people who've experienced it to study properly. There's something fitting about ending the more optimistic, hopeful thread of this research, right where the whole story of human spaceflight technically began, with someone simply looking back at the planet they came from. Every other finding covered in this video traces back to some form of loss: the loss of gravity's steady pull, the loss of Earth's magnetic shielding, the loss of a normal circadian rhythm, the loss of ordinary daily social contact. The overview effect is the one major finding that traces back instead to a genuinely new kind of perspective on the world that simply wasn't available to any human being before spaceflight made it possible to physically leave the planet and turn around to look back at it whole. Whatever the eventual neuroscience turns out to say about exactly how that particular gain happens inside the brain, it stands as a useful counterweight to everything else in this story, a reminder that the same environment capable of measurably reshaping and stressing the human brain also appears capable of expanding it in ways nothing on Earth has ever quite managed to replicate, and that whichever side of this story ends up mattering more to any individual reading the research, the damage or the growth, both are equally real, equally documented, and equally worth understanding before humanity commits to sending anyone that far from home for that long. Hold both of those realities at once as we head into the final wrap-up, because that's really the accurate, fully honest version of this story. Not a warning and not a promise, but a genuinely mixed, evidence-based picture of a brain built for one planet, quietly learning, adapting, and occasionally struggling as a small number of human beings have begun asking it to function somewhere else entirely. One MRI scan, one blood sample, one carefully logged night of restless sleep, and one long, quiet look back at Earth at a time. Before wrapping up, it's worth taking stock of exactly where all of this research actually stands right now, today, rather than treating any of it as a finished, closed case. As of the most recent published research available, SANS remains an active, unsolved medical priority for NASA, with expert consensus papers still being published in an effort to standardize exactly how it should be diagnosed and managed across different space agencies. NASA's 14-study CIPHER investigation is still ongoing, deliberately designed to extrapolate what's been learned from shorter ISS missions toward the much longer timeline a real Mars mission would require. And perhaps most importantly, the single biggest gap in this entire body of research remains exactly what it's been from the start. No human being has yet undertaken a true multi-year mission fully outside Earth's protective magnetosphere. That means essentially all of the most severe radiation-related brain damage findings covered in this video still rest on rodent studies and mathematical extrapolation, not direct, observed human data. That's not a criticism of the science. It's simply an honest description of where the evidentiary line currently sits. And it's exactly why so many different research teams across multiple countries and space agencies are racing to close that gap through better shielding, better pharmaceutical countermeasures, and better predictive modeling before any space agency commits to actually sending a crew on that trip. Passive shielding, active electromagnetic shielding, and pharmaceutical neuroprotection are all still being actively developed in parallel. And as of right now, no single one of those three approaches has been finalized as the adopted solution for an eventual Mars-bound crew. Whichever combination of these approaches eventually wins out, the underlying research covered throughout this video, the twin study, the rodent radiation trials, the MRI fluid shift findings, the SANS consensus papers, and the isolation research from Mars 500 and HICs will form the actual evidentiary foundation that decision ends up resting on, long after this particular video has been watched, discussed, and eventually replaced by whatever the next round of published findings ends up showing. So where does all of this actually leave us? We started this video with a simple, direct claim that deep space quietly rewires the human brain. Having walked through the actual published evidence, that claim holds up, though the real picture turns out to be considerably more layered and more interesting than a single simple headline could ever capture. Scott Kelly's year in orbit gave researchers their first detailed human dataset, showing measurable chromosomal change, immune system disruption, and a genuinely mixed, honest picture on cognition. Some domains affected, others apparently untouched. Charles Limoli's rodent research and the parallel work coming out of Eastern Virginia Medical School points toward a genuinely serious radiation risk still unresolved for any future Mars-length mission, built on solid animal data, but still, by the researchers' own admission, an extrapolation rather than a confirmed human outcome. Meanwhile, an entirely separate and already well-documented body of MRI research shows that simply removing gravity itself, no cosmic radiation required, physically reshapes the astronaut brain through fluid shifts, ventricular expansion, and measurable gray and white matter remodeling, changes that are real, replicated across multiple independent studies, and in some cases take up to 3 full years to fully reverse after return to Earth. Layered on top of all of that sits SANS, a common and still not fully understood syndrome affecting a genuine majority of long-duration astronauts. Chronic sleep deprivation that chips away at specific cognitive domains even when overall performance looks stable on average, and the well-documented psychological toll of true long-term isolation demonstrated across Mars 500, HICs, and decades of parallel research in Antarctic and polar environments. And running through all of it, seemingly independent of every one of those physical stressors, sits the overview effect, a profound and remarkably consistent, positive psychological shift that researchers are still working to fully understand at a neurological level, even as its real-world consequences on astronaut attitudes and behavior are already well documented. What all of this ultimately points toward is not a simple story of space damaging the brain, nor a simple story of space transcending it. It's a story about how deeply, intricately connected the human brain is to the specific planetary conditions it evolved inside of: gravity, a protective magnetic field, a stable day-night cycle, an environment allowing normal social contact, and what happens, measurably and in some cases quite dramatically, once you start removing those conditions one at a time. Every scientist quoted in this video is working, right now, on some piece of the puzzle required to eventually solve this. Whether that's Charles Limoli chasing pharmaceutical countermeasures against radiation damage, Richard Britton trying to understand exactly what's biochemically broken so it can eventually be fixed, or the combined NASA and ESA teams behind Cypher, neuromapping, and Brain DTI, quietly gathering the data that will eventually decide whether, and how, and under what protections, human beings finally make that 2 to 3 year trip to Mars, with their brains and their minds arriving intact on the other end. There's something worth sitting with in the fact that so much of this research is happening simultaneously across so many different institutions and countries. University of California, Irvine, Eastern Virginia Medical School, NASA, the European Space Agency, and Roscosmos, all working different pieces of essentially the same underlying puzzle, often without any single team holding the complete picture on their own. That's genuinely how a lot of real, unresolved science works. Not one dramatic breakthrough moment, but a slow, patient accumulation of MRI scans, blood samples, rodent behavioral data, and carefully logged sleep diaries, gradually building toward an answer nobody has yet fully assembled. The twins study alone took 27 months of sample collection and years more of analysis before its results reached publication. The scientific report study on ventricular plateauing needed astronauts followed for up to 3 years after landing just to capture the full recovery timeline. Mars 500 required 520 consecutive days of 6 people simply staying inside a sealed facility together before researchers had anything close to a complete psychological dataset. None of this moves quickly, and none of it is designed to. It's designed to be right, which, when the eventual outcome is whether real human beings can safely spend 2 to 3 years of their lives outside the protection of everything our species evolved inside of, seems like exactly the right priority to hold on to. It's also worth remembering that every single piece of this evidence, the chromosome data, the shrinking dendrites, the expanding ventricles, the sleep logs, the isolation questionnaires, and even the astronaut testimony behind the overview effect, exists because a relatively small number of people volunteered to be studied this closely in the first place. Scott and Mark Kelly agreeing to years of blood draws and cognitive testing. 6 Mars 500 volunteers agreeing to disappear from the outside world for 520 days, 13-minute communication delay and all. High seas crews agreeing to live inside a dome on a Hawaiian volcano for the better part of a year, working through conflict and stress modules on a laptop, because that was the only mental health support available to them. Every astronaut who let researchers scan their brain before launch, and then again months or years after landing, tracking a recovery process that in some cases outlasted the mission itself several times over. None of what's covered in this video would exist without that willingness, and it's worth acknowledging plainly before closing out. If you made it this far, thank you for spending this time genuinely learning something real about what space travel actually does to us, rather than settling for the usual highlight reel version of space exploration. This is a story that's still being written in real time by researchers at UC Irvine and Eastern Virginia Medical School running the next round of radiation trials, by NASA and ESA teams pulling fresh MRI scans off returning ISS crews every few months, by the Mars 500 and HIC's data still being reanalyzed years after those crews walked back out into daylight, and by every future astronaut who volunteers to be measured, scanned, and studied this closely, so that the people who eventually do make that 2 to 3 year trip to Mars have a genuinely better, safer, more informed understanding of what their own brain is about to go through. If any of this surprised you, or if you've got your own thoughts on whether you'd personally take that trip knowing everything you now know, I'd genuinely love to hear it in the comments below.